Welcome to Folds and Curves. I'm your host, Mokash Kush. Today's awesome guest is an evolutionary biologist and microbiologist. Vaughn Cooper is a professor of microbiology and molecular genetics and computational and systems biology at the University of Pittsburgh School of Medicine. He's co-founded and is director of the Center for Evolutionary Biology and Medicine, founded and directs the Microbial Genomics Sequencing Center, and is the chair of the Council for Microbial Sciences for the American Society of Microbiology. His research is supported by the NIH, NSF, NASA, and the Cystic Fibrosis Foundation. And the Cooper Lab focuses much of their work on how bacterial populations evolve, especially with respect to biofilms and antimicrobial resistance, as well as why genome regions evolve at different rates. On this episode, we discuss some of the evolutionary biology concepts that apply to COVID-19 variants, as well as EvolvingSTEM, which is an initiative that Vaughn is passionate about, which engages high school students with topics in evolution and heredity by conducting a week-long experiment with bacteria. This episode was really informative for me, and I hope it will be for you too. Make sure to check out the show notes to learn more about Vaughn. With that said, let's get started. Hi, Vaughn. Thanks so much for joining us. I wanted to start with asking you about variants and strains and how they relate to, say, just a mutation in a virus. I've gotten this question a lot from people who aren't in medicine or the life sciences. And I always start with that viruses are always mutating. But beyond that, I feel like I don't have a clear answer for what is actually a strain versus a variant.
Yeah, thanks, Mo. This is a good. Thanks for asking, and thanks for having me. So I think it's still, we are in what, month 16 or something like that of the pandemic, and I think the official line really should be that there's still only one strain of the novel coronavirus, SARS-CoV-2. And the reason I think we can say that is really how we define mutations and variants, kind of technically. So what we do is we sequence fragments of the virus and stitch them all together. But the way we do that is by mapping every one of those reads of nucleic acid sequence against the original reference. And that continues to work. So I know that's a very technical answer, but what it means to say is that there really is only one parent of all of these viruses, and all of them are no more than 30 to 40 mutations, maybe a few more in some weird conditions, different from that first original ancestral Wuhan 1 isolate that was sequenced and released in early January of 2000. And so, that's a kind of a long answer to your question where there's one strain. Any two viruses that you grab from around the world right now, call it month 16 of the pandemic or month 15, would differ by about 30 mutations, 2 mutations per month on average, drawn at random. Now, if you had 2 from the same outbreak, they may differ by 0 mutations. And then, so that's what just sort of mutations. Now, variants really are just particular combinations of mutations that appear to be rising in frequency, or actually are. So the well-reported UK variant, also called B.1.1.7 or B117, that's a combination of mutations that defines a lineage that is definitely more fit. It's more transmissible, and it is probably also a bit more severe. So those are 3 different answers to your question. There's one strain, there's lots of mutations, and there's only, a few handfuls of variants that are just lineages that are, as combinations more fit than the original Wuhan 1 ancestor.
Yeah, thanks for answering. I kind of want to dive into this word you used, fit, and I guess I'm curious, what traits are important for fitness in a virus? Is it transmission? Is it how deadly it is? What traits make a virus sort of rise in prevalence in a certain area?
Yeah, it's all about transmission,
Okay.
right? So a virus that is very, very good at reproducing within one patient, but and reaches extremely high levels in that one patient, but fails to transmit, is a very unfit, basically fitness zero
Mm-hmm.
lineage. So, and likewise, there are other viruses that actually don't reproduce a lot, but transmit really well. Those are more fit. And so the endemic coronaviruses that have been with us for a long time, they're quite fit. They cause a lot of annoying colds, and they bounce around with essentially zero interference. This virus is actually quite fit. It arrived on the scene with a tremendously high probability of transmission, but that's going to change as immunity builds. And so then the real question is, what happens next?
Okay, so I'm kind of glad you brought up common cold coronaviruses, because my understanding is most of us get infected with these as children, and then we don't really get too sick, and then we sort of develop this very weak immunity towards them
Mhm.
throughout our life. So as we build up immunity with COVID or SARS-CoV-2, either through vaccination or through infection, um, would you, as an evolutionary biologist, suspect something similar to happen with this particular coronavirus, or is it kind of impossible to say?
Well, I think somewhere in between. I mean, I don't think we can certainly make a reasonable forecast.
Mm-hmm.
The reasonable forecast is that it actually comes from what we know about our experience from a more closely related coronavirus, about its duration of immunity. And so that's with the original SARS. So although we don't have that many patients to follow from the original SARS coronavirus, patients that were infected back in the early 2000s, 2003, 4, with that pandemic, still show some lasting immunity to SARS-like coronaviruses. So that suggests that there's actually pretty long-lived general immunity, which would be somewhat protective. And that's different from these sort of cold-causing coronaviruses, which really they do have different ancestry. So these coronaviruses, these SARS-like coronaviruses, come essentially directly from bats, maybe through an intermediate host, but they're generally bat viruses.
Mm-hmm.
The endemic coronaviruses are actually circulating in livestock and in other animals other than bats. And that could be why they prompt a weaker, they're less severe, prompt a weaker immune response. It could be because there's a longer history of coevolution between us and them in the interim that we just haven't resolved. So I don't know. I mean, I remain cautiously optimistic that, I think we're stuck with SARS-CoV-2 for the long haul. I want to set that out too. It's not going away, but I think I'm hopeful that it'll be tamped down quite a bit rather than becoming this, sort of weak and annoying cold-like virus. Maybe it will. Who knows? It's hard to say, truthfully.
Yeah.
But it does seem that the immune response against this is pretty strong,
Sure.
and durable.
Mm-hmm. You brought up the mutation rate earlier. I kind of wanted to dive a little bit deeper into that. How similar is that to, I guess, one, other coronaviruses, and two, other RNA viruses?
Yep. Great question. So it's very similar to other coronaviruses, which makes it one of the best RNA viruses at reproducing and taking care of its genome because it's got this proofreader. At the overall level of the genome, it trades off a little bit because it's actually, for an RNA virus, a relatively big genome. So there's just sort of more to replicate, and you know, the more you replicate, the more errors you make. But it's still several fold better at sort of high fidelity replication than another more commonly studied RNA virus, which is the influenza, or other RNA viruses for that matter. Their mutation rates tend to be 5 to 10-fold higher because they don't have that proofreader enzyme. And that I think is another reason why it's still easy to call this all one strain. It's still in the process of this steady diversification from one common source, and it's not split off into wild and different lineages. It also doesn't recombine, so that's another key point. So it's not just the mutation rate, but it's whether segments of the virus can mix and match. So that you know, flu has a segmented genome, and that allows for reassortment, which leads to basically quantum changes in those strains, and that has not happened for this coronavirus, and we expect it to be very rare for coronaviruses. It does happen in rare instances, but it's quite rare.
Yeah, so those sort of like dramatic genetic changes with flu, that's sort of why we have yearly vaccinations, right? Because of how dramatically it can change, or is that not correct?
Yeah, so I mean, it's so the reason why we have to have flu boosters is a product of a few things. The first is there are a lot of flu lineages that are circulating, and they also mutate a bit more, and occasionally they reassort or recombine and make new combinations. I think the big reason why we have to re-engineer the vaccine every year is because, again, just the standing diversity. The rare strains tend to be, if you are rare this year, you are going to be more fit next year, because most people have not developed immunity against you because you're rare. So there's this continual advantage of being rare. And so once you go from rare to common, then you're not as fit anymore because there's more immunity against you, right? And that's also boosted by the fact that we provide vaccines that train many immune systems against that variant. So, you know, you can basically bet that the virus that was part of the cocktail for the vaccine last year will be unfit the following year because there's so much immunity against it. So you have that, then they mutate more. And so sometimes those lineages, even though they might have been part of a prior vaccination campaign, evolve the ability to escape prior immunity by mutation. And then you have the third factor, which is less common, but more concerning, which is basically the recombination of existing variants, both circulating in humans and also circulating in fowl, you know, ducks and pigs, that literally make essentially evolve new viruses in combination. And then there's literally no immunity against those, and those require more immediate action, if that makes some sense. So yeah, no, that layered problem. And I think like all of that is eventually going to happen with this coronavirus too, but it's going to take a long time to establish multiple durable lineages that, you know, that can each have the potential of escaping existing immunity.
Okay. And you think it'll take a longer time? Is it partially because of the mutation rate or partially just because it's so rare still? Well,
It's well, I mean, yeah, so it's not rare. It's like
sorry,
super
I mean
common,
like,
right? But it's all one thing. So
mhm.
so it's a combination of the mutation rate, and because we are just starting to get to sort of the next phase of the evolutionary process, which is immune evasion, right? So right now we think we know about really only one mutation, one particular change that that can escape prior immunity, and that's E484K and spike. That mutation has been shown to contribute to the evasion of prior immunity. And then there are some other mutations that may, may move the needle in that regard too, and the combinations there are less clear. So that's
Okay.
just the beginning of that process.
Gotcha. So I got kind of curious about when you mentioned, a virus being rare at some point in its life. And I guess like, how do we, this is more of like an operational question. It's like the global surveillance community. How do we actually keep track of what's rare and what's common? And maybe if it's genetically different than another. So like, how do we keep track of different SARS-CoV-2 variants even?
Yeah, so I mean, I think you've probably heard me argue that we need to do a better job of genomic surveillance. So not just testing, but actually sequencing the entire, 29,000 plus nucleotides of the virus and looking for those variations. We need to do a heck of a lot better job of that in the United States. And I think that there's momentum and some bills in Congress building to support that. And that would be more broadly, that's an investment in public health infrastructure. And also in the know-how, the analysis know-how to interpret that data. So I always tell folks that, sequencing is effectively cheap, analysis is expensive. And our training, the way we teach, and the way we recruit and the way we budget for grants just does not reflect that. And we as a broad scientific community have to change that. We have to teach our students to analyze the data. We have to budget for our grants, and we have to write that into bills, which say we need to invest in training, public health officials, workers to become more self-sufficient with this data.
Gotcha. So when you say analysis, is this referring to like hiring people and training people, training graduate students, postdocs, staff?
Yeah, and yes, it's about training undergraduates about working with data. One of the things I'm really excited about is our EvolvingSTEM, evolution and action curriculum for high school students, where students learn to become a little bit literate with the big data associated with genome sequencing and to identify mutations. Like, there's no reason why we can't do that as part of all life science education. Genomics is current, and it's becoming cheaper and more easy to access. So we want to build that capacity, so that if a student wants to become part of the public health solution, they can.
Yeah, I'm kind of curious about EvolvingSTEM more. I know it's a little bit off the COVID topic, but I did see a video on your YouTube channel from Winnicott High School, where one of my friends went to high school. So I'm kind of curious, what are you teaching high school students? Are you giving them, are they actually sequencing data?
Yeah, so
thanks.
Genomes? Yeah. So they themselves are not sequencing the genomes, but so yeah, we're in about 12 or 13 high schools around the country, and we've been working hard to give remote, students now working remotely some new experiences in that. And over the last year, we've really, 6 months or whatever, we've really invested in making the bioinformatic experience accessible to the students. And so to answer your question, I put a little bit more perspective. The students do an actual bacterial evolution experiment, right? So they start out with a single clone of bacteria, they passage it for a few days, selecting for the bacteria to form a biofilm on a plastic bead. And by the end of the week, mutants have been selected that look different. So they can see a phenotypic change in the bacterial population over the course of a week. And so that's usually where the experience stops. They notice that evolution happens, and then they learn that that is a product of natural selection acting on new mutations, where the better the biofilm former, the more fit that variant, and the more likely you'll actually see it. But the natural question is, what's the gene or the mutation that is responsible for that? So what we can do is have them pick their favorite mutant, send it to us. We can extract the DNA from that mutant and then sequence the complete 6,000 plus letters of the genome and send a file of all of those reads to them. And that's when the experience starts. They can sort of download that data and then do exactly what we do with the novel coronavirus. You just map all of those reads against the known ancestor, and you find out exactly, where, if anywhere, there's a difference. And usually there's one or two differences in genes that underlie how the bacteria sort of balances the switch between a swimming and sticking life cycle. So yeah, I mean, it's the real deal. As I say, it's kind of pro-level bioinformatics, the kind of bioinformatics that a lot of my colleagues ask us to do for them. They can do it themselves. I'd say you just do the EvolvingSTEM experiment and you'll become bioinformatics literate yourself. I hope that answers your question.
No, that's really, really cool. I never had anything like that when I was in high school, which is
me neither.
Yeah.
I mean, it wasn't a thing when it wasn't a thing. And
it couldn't have been. But like, for example, my younger brother is this, junior and now in high school. When I was in high school, they also didn't have computer science classes really. And now he knows how to program, and he's only 17. Basic level. But I think it's sort of somewhat of a similar trend probably. I would hope with bioinformatics and genetics.
I think I'm hopeful, but I think we have a ways to go to I guess I'll call it build the bridge between the data science or the analysis methods that we use for biology with computer science or other kinds of analytics. I mean, it really is kind of stepping back and thinking about what actual research experience do you want to weave into the teaching? So, I'm a huge advocate for learning by doing. And that's going to start with allowing, basically engaging the teachers in that process too. So, we're at the point where the students learn to pick up these curricula a lot faster than the teachers do. You've probably noticed that with your brother, right? So, I mean, and I don't expect, and this is true, we have wonderful high school teacher partners, but this bioinformatics stuff is kind of scary because it's, big data and maybe even require using command line. And, I mean, that's just not what most of us have been brought up doing. I've been really lucky to kind of have had that around me most of my life, but I know that that's unusual. So it is a cultural thing. And then, obviously we want to continue to support programs that support tech development, and again, learning by doing rather than kind of textbook learning.
Okay. Yeah. I think learning by doing's, definitely for me always been more beneficial. I always seem to forget the things I read eventually.
Right. Yeah. And I think, I think ultimately what you want to do is you're not going to remember exactly how to do this all yourself, but if you're given a reusable roadmap to do it again and fumble your way through the directions on your own, then then you might wind up finding yourself actually doing bioinformatics for somebody else sometime.
Yeah. So, in the experiment the students did or do,
Yep.
you mentioned a reference genome. You also have mentioned it in reference to our earlier conversation about COVID. And I guess I was sort of preparing for this discussion a little bit, learning about evolution and virology. And I came across something called quasi-species theory.
Yeah.
And it was, if I'm understanding it right, it was kind of like there's no such thing as one reference genome, and it's sort of an average almost of.
Yep.
And so could you kind of like unwind that a little bit, or did I do a good job?
No, you did a nice job. And I think that you brought up a super point. So, I mean, I think if you capture all of the coronavirus sequences, basically, if you could sequence every virus that's currently in a human right now of the novel coronavirus, you would have a giant quasi-species. But to bring it more locally, even within a patient, particularly say an immune compromised patient where the infection might last a bit longer, that virus just by replicating is going to make errors. And before you know it, it produces a cloud of variation. So maybe we should focus on that to answer the question. If you look at a long run, sort of poorly contained infection in a patient who's not clearing that infection for whatever reason, it becomes a population, a quasi-species, which is really just another way to describe genetic variation, right? I mean, you could sort of scaling up to us. I mean, our two genomes are really different. The one from your mom and your dad, they're different. And the amount of variation, in a classroom when we get back together is pretty substantial. And the amount of variation in our entire species is pretty substantial. But, actually, the overall genetic variation in Homo sapiens is a lot less than you might think, because we all basically trace back to, very few numbers of genomes a few 100,000 years ago. So effectively, we're still almost clonal. And I think this is I really wish we would talk about this more in our classrooms and in our lectures. We're not as different from one another as we think. And yet we really tend to overemphasize that genetic variation or that ethnic or social variation. There is no such thing as wild type human. There is still maybe wild type SARS-CoV-2, but really there isn't, because you're not going to find that virus anymore. It's gone. So I think, at this point, all we have are lots of descendants of that original one. You don't see the original Wuhan 1 strain causing a new infection anymore, not in the nowhere in the world, not unless it was released from a freezer stock, right? It's gone. Yeah. It's history. Same sort of situation with all species, right? There is no such thing as wild type. So I think quasi-species is a fun concept, but I think all it is just a snapshot of genetic variation, and sort of the scale of that cloud of variation depends on how old the population is and how much what the mutation rate there is. I know it kind of said a lot, but I think we all need to embrace genetic variation. Is what I'm kind of saying.
Yeah, let's do it. I'd be curious maybe offline to see if there's any way to do that through the education you do with high school students, if there's a way
Thanks.
to actually use biology as a way of sort of teaching a better social structure or way we interact with each other. But we'll save that for offline. I'm curious about the point about the original SARS-CoV-2 not being around. I didn't know that. That's news to me. That's pretty interesting. Is this just, as you would expect with sort of basic evolution, it's not that fit compared to some of the descendants? Is that why?
Yeah, so it's funny, it's been basically undetectable for, probably since like March of 2020, not 2021, right? So
Jeez.
it was replaced basically by chance, really early on, because as sort of mutations happen and lots of lineages, basically most lineages go extinct, right? So most mutations in general in all species, and especially in viruses that go through these very small bottlenecks in their from transmission, most lineages go extinct. Most of the mutations, in fact, essentially all mutations on average that arise within a coronavirus infection don't make it to the next host. So that process of random sampling, which is basically genetic drift, causes old sequences to go away. And it wasn't until really this summer when the first truly adaptive mutation, this D614G variant, started to rise and really take over the world. It wasn't until that sort of sweep of a variant that we can say there was adaptation to humans. Before that, it was just all random sampling, and the sequences of the viruses were super for genetic epidemiology, but they were useless for our understanding of adaptations. But even still, that randomness caused the original Wuhan 1 sequence to just disappear. It went extinct by chance. Yeah, so I know that sounds complicated, but this epidemic has been an unbelievable lesson for how evolutionary biology works. This is rewriting our evolution curricula. We have convergent evolution, right? We have drift, we have, natural selection, we don't have recombination, thank God, but we can talk about it.
Yeah. So with the adaptation, with that specific one you mentioned, is that in the, I assume it's in the spike protein because that's sort of what you always hear about, but
yep.
So is it just better binding to ACE2?
It is.
Okay.
Yeah. Yeah. So D614G has an ACE2 binding advantage. It's modest, but it's enough to improve the lineage fitness by about 20%.
Okay.
And that's a lot, right? So it is now not more than 90% of viruses around the world have the G at 614 instead of the original D. Interestingly, in the remaining 10%, there's other lineages that have 614N instead of D. So there's an instance of site-specific convergent evolution of new changes at that site that are more fit than the original aspartate.
Gotcha. Huh. So what about sort of dodging neutralizing antibodies? I mean, thankfully, it sounds like with the vaccines that they all still, from a clinical perspective, have a protection against severe disease that I've seen so far, at least.
Yep.
But
yeah, these are miraculous, right? I mean, home run. Yeah.
Yes. But I have seen some of the neutralizing antibodies differ between the variants, and I'm just kind of curious if you could give an evolutionary backdrop to why that is. What is the virus trying to do there? Is it, and maybe like mechanistically, is it changing the structure of the target of that antibody to make it not be able to bind to it, or is it just completely maybe I'll just let you explain.
You know what, Mo? You've reached the end of my depth. I'm a lousy a lousy immunologist, and I should say I'm only you know, I'm a very passing sort of epidemiologist. So how is it that the immune responses vary to these different vaccines?
Or sorry, one vaccine, different variants, different.
Okay, one vaccine, different variants.
Yeah, creating different levels of antibodies.
So I don't know. And honestly, I'm not sure how well it's even known yet. So I get this question a lot, from folks asking about how these, you say, Vaughn, you say that this variant has a set of adaptations. How do you know that they are adaptations, and how do they work? And I say, I know that they're adaptations because they're all becoming more frequent at once. Like, that's the finding. That is by definition more fitness, because they are reproductive, they're transmitting more, and their numbers are going up. We can see that in the data, but how does that work? Gosh, I don't know. Others may know, and I have to plead some ignorance. Maybe it's known and it's been published, and I haven't been up on that literature. But more often than not, these days, we can define things as more fit quicker than we can figure out how they are more fit, how they evade immunity, or how they bind, because that's like tricky cell biology. It's slow going. So I'll give you an example. For the variants that the group that I'm a part of discovered at position 677, they've become 3 times more frequent since we first published the preprint about this about 3 weeks ago, in the overall sampling, whereas the total number of samples has only gone up about 50%. So 3 times versus 0.5 fold, that's a lot, right? That's a significant increase in frequency, and it's an unbiased monitoring effect. So there's no bias in sampling. So by definition, it's more fit, right? Multiple independent lineages, all increasing in frequency. Then people say, how does it work? And I say, I don't know.
Gotcha.
I can say that it's 4 residues away from a site that we know is super important for the function of spike. But how does it work? I mean, that's the hard part. So then you put together a team that has the expertise to basically do a competition experiment between the old virus and the new virus, mix them together and see how they win, who wins under different conditions, and whether neutralizing antibodies have anything to do with that fitness or not. That's the beginning of the study. That's slow and expensive science. Super important, but it's hard.
Yeah, no, I really appreciate how candid your answer is. It actually helps me sort of understand what you're working with. It sounds like you're working with, the frequency is increasing, and you know where the mutation is.
We do,
and that's actually very helpful for me to understand what you look at, what data you're looking at.
Yeah, so I mean, I think the point I always want to say is that what evolution is doing for us is narrowing our focus to avoid wasted energy on sites that are unimportant for the current state of play between us and it. And that's true for the bacteria that we study. We study how antimicrobial resistance evolves. Well, there could be lots of genes that could be involved, but when you see changes in the same gene and even at the same position happening over and over and over again, that convergent or parallel evolution really allows us to zoom in and say, okay, we need to figure out how that works, that one change. And then you build a team around you to answer it.
Awesome. I just wanted to know, is drug resistance a problem with COVID, with the antivirals? So like, I'm thinking about remdesivir here.
Yeah, so there's been some, you can talk about this, put this in. So there's been some interest in studying that, particularly in the patients who have had sort of almost chronic COVID because of being immune compromised. So people who are immune suppressed because they're on chemotherapy. They've been treated with remdesivir and other antivirals and therapies. And so there's been some speculation that the long branches, those really more divergent viruses, may have mutated. Well, they definitely mutated, but those mutations might have been selected in part for their ability to avoid the antiviral. That's speculation. I've not seen any direct test of evolved resistance, but you might, I mean, there's the selection's strong. It seems very plausible that that could happen. I think we're still a little bit limited by sample size to say anything convincing about that yet.
Hey everyone, thanks for listening. I really learned a lot on that episode. I hope you did too. Don't forget to check out the show notes. And with that, take care.